Welcome. Today, we're diving into one of the most incredible pieces of biological engineering in the entire human body, the inner ear. We're going to really unpack the secrets of how this amazing structure actually makes hearing possible.
All right, so just to lay it all out, this is the first of a two-part series. To really get how we hear, we first have to build the inner ear from the ground up. So in this segment, we're focusing on the architecture, basically building the power supply. Then in part two, we'll plug everything in and see how the organ of Corti uses that power to do its magic.
And that really gets us to the core question, doesn't it? Sound waves. Well, they're just mechanical vibrations. But our brain? It only understands one thing: electrical impulses. So the inner ear has to be a transducer, something that converts that mechanical energy into electrical signals. And just like any good piece of electronics, it needs a battery. So how on earth is this biological battery built?
Well, we're going to start with the outer casing of our battery, this truly remarkable structure known as the bony labyrinth.
Think of this thing as the ultimate fortress. It's this incredibly complex network of tunnels carved directly into the petrous temporal bone, which, by the way, is the densest bone in your entire body. Its whole job is simple: provide a rock-solid, super stable, and protected home for the unbelievably delicate machinery that's tucked away inside.
Okay, so nestled inside that hard, bony casing is the active component, the real functional core of our battery, the membranous labyrinth.
Now, this is a much more delicate, flexible system of sacs and ducts, and it's just floating inside the bony labyrinth, almost like a ship in a bottle. But make no mistake, this is where all the action happens. This structure houses the actual sensory hardware that makes hearing possible in the first place.
So here's the key takeaway, the big contrast you really need to get. You've got the hard, bony labyrinth acting as the protective shell, and floating inside it, you have the soft, membranous labyrinth, which is the functional core. And notice they each contain a totally different fluid. Now, that might seem like a minor detail, but trust me, it's the absolute secret to the entire system.
The difference between these two fluids is literally the key to the inner ear's power source. It creates a chemical and electrical gradient that is totally unique in the entire body. So let's take a closer look at how this all gets set up.
First up, we have perilymph. This is the fluid that fills up that bony labyrinth and bathes the outside of the membranous labyrinth. You know, biochemically, it's pretty standard stuff. It looks a lot like the fluid you'd find outside most of the cells in your body. It's high in sodium, low in potassium. Nothing too exotic.
But then there's endolymph, and this is where things get really fascinating. This fluid, which is only found inside the membranous labyrinth, is unlike anything else in the body. Its ionic makeup is basically a mirror image of perilymph. It is packed with potassium and has very, very little sodium, making it look a lot more like the fluid you'd find inside a typical cell.
Now, just look at how different these are. You've got two fluids separated by just a paper-thin membrane with this huge difference in ion concentration. But the most incredible part? Check out the electrical potential. The endolymph isn't neutral. It's carrying a powerful positive charge of about 80 millivolts. And that begs a really important question, right? Maintaining a steep electrochemical gradient like this is like trying to hold back a flood with a leaky dam. It takes constant work and a ton of energy because everything in nature wants to just even out. So what's the biological power plant that's constantly working to maintain this incredible charge?
The answer lies in this highly specialized, metabolically super active tissue that lines the outer wall of the cochlear duct. It's a structure called the stria vascularis. The stria vascularis is an absolute metabolic furnace. It has one of the highest rates of blood flow of any tissue in the body, and that just reflects its nonstop job as a biological pump. Its entire existence is dedicated to doing one thing: actively pumping potassium ions into the endolymph to generate that powerful positive charge.
Now, to keep this incredibly precise and delicate chemical balance, the whole system has to be sealed off from the rest of the body. And that's the job of the blood labyrinth barrier. You can think of it like a high-security checkpoint. It uses these really tight cellular junctions to make absolutely sure that the unique composition of the endolymph isn't thrown off by even the tiniest fluctuations in your blood chemistry.
And you can see the whole process laid out right here. First, that barrier creates this isolated, perfectly controlled environment. Then, these specialized cells within the stria vascularis get to work. They use active pumps to literally force potassium into the endolymph, pushing it against its natural gradient. And it's this relentless pumping that builds up that incredible electrical potential.
So, after all that complex anatomy, all that intense metabolic work, what's the final result? What's the big payoff?
This, a positive 80 millivolt electrical potential. That is the official voltage of our cochlea battery, a charge that is completely unique in the entire human body.
And here's why that number is such a game changer. That plus 80 millivolt potential, combined with the negative charge that's naturally inside the sensory hair cells, creates this massive electrochemical gradient of about 150 millivolts. The system is basically a loaded spring, just packed with stored energy, making those sensory cells unbelievably sensitive to even the absolute tiniest sound vibration.
So to wrap it all up, the inner ear isn't just a passive microphone waiting for sound. No way! It is an active, self-powered biological battery. It is constantly being charged by the stria vascularis to create what's called the endocochlear potential, a massive store of energy ready and waiting to convert the mechanical whisper of sound into an electrical signal the brain can finally understand. And that raises the next big question, doesn't it? How is this plus 80 millivolts of pure potential energy unleashed by the faintest of sounds? We'll get into that in part 2. And by the way, if you like these deep dives, please go ahead and like and subscribe for more custom medical videos made just for you. Now, moving on to the next segment.
All right, so picking up right where we left off, let's continue with our deep dive into the inner ear. Now, we're zooming in on the main event, the organ of Corti and its incredible micromechanics. Seriously, this is where all the magic of hearing really happens.
Okay, so we're moving past the big picture anatomy and we're going right into the microscopic engine room of the cochlea. This is the biological stage where the raw physical energy of a sound wave gets translated into the electrical language that your brain can actually understand. So let's get a closer look.
So what is the organ of Corti? Well, the easiest way to think about it is as the biological microphone of your inner ear. It's this beautiful, intricate strip of sensory cells that does one absolutely critical job: transduction. Without this process, all those vibrations we've been talking about would just be meaningless noise. This is where the symphony of sound gets written for the brain.
Okay, let's meet the players in this little biological drama. We've got our superstars, the inner and outer hair cells, or IHCs and OHCs. But they're not working alone, not by a long shot. They have a whole supporting cast, cells like the pillar and Dieter cells, that give the whole structure its shape and strength. And hovering over all of it is this gelatinous canopy called the tectorial membrane. The whole thing is a precisely organized, living machine.
And check this out, running right down the center is this space called the tunnel of Corti. Now, it's not just some empty gap. It's a really important structural feature formed by those pillar cells we just mentioned. It acts as a crucial divider, separating the whole organ into these distinct inner and outer sections. And that separation is key to how the whole system works.
So this all brings up a really fascinating question, right? If the goal is just to detect sound vibrations, why would evolution go to the trouble of creating not one, but two totally different types of hair cells? I mean, it kind of seems redundant. But as we're about to see, they have dramatically different and equally vital jobs to do.
Wow, okay, this table really tells an amazing story. On one side, you have the inner hair cells. There's just a single row of them, but look at this. They are connected to 90 to 95% of the nerve fibers that go to the brain. These are the cells that do the actual hearing. Then you've got the outer hair cells. There are way more of them, 3 whole rows, but they're barely talking to the brain at all. So you have to ask, what on earth are they for? Well, look at their function. Cochlear amplifier. And that, my friends, is where things get really cool.
So here's the big reveal, the game changer. Your cochlea isn't just a passive microphone that sits there and listens. No way. It is an active system. It has its own built-in high-performance amplifier. This discovery completely flipped our understanding of hearing on its head. And the engine driving that amplification? You guessed it. It's those mysterious outer hair cells.
They do it using a kind of biological superpower called electromotility. It sounds like something from a comic book, right? It's this incredible ability to physically change their length, to get shorter and longer at mind-boggling speeds, all triggered by tiny electrical signals. And this isn't like a muscle flexing. It's way, way faster. It's all powered by a unique motor protein called Brainstem, which is just jam-packed into the walls of these cells.
Let's walk through the cycle. So a sound vibration comes in and causes a tiny electrical change in the outer hair cell, a depolarization. That little zap is the on switch for millions of Brainstem motors, which instantly change shape, causing the whole cell to shorten. Bam!
Because the cell is physically attached, this shortening tugs on the basilar membrane, adding energy back into the vibration at the perfect time. They are literally dancing in sync with the sound, pushing and pulling to make the vibration bigger and bigger.
And the effect of this is staggering. This active process, this cochlear amplifier, can boost the sound signal by 40 to 60 decibels. To put that into perspective, that's the difference between a sound being an almost inaudible whisper versus a clear, easy-to-hear voice. This process juices up the signal, giving the inner hair cells something clean and powerful to detect.
This quote just nails it. A biological piezoelectric amplifier. It's a very technical term, but it basically means it's a piece of natural nanotechnology that turns electrical energy back into physical movement with incredible speed and precision. It is, quite simply, an engineering marvel, and it's happening inside your head right this second.
But here's a really crucial point. This isn't just about turning up the volume. The real magic here is that this amplification also sharpens the tuning of the cochlea. It's like focusing a blurry image. It takes a broad, kind of messy vibration and hones it down into a razor-sharp peak. And that is what lets us distinguish between two very similar musical notes or understand the subtle differences in speech.
This whole idea of frequency tuning leads us to another fundamental principle of the cochlea, tonotopic organization. It's a big word, but the concept is actually pretty simple. It just means there's a frequency map laid out along the basilar membrane. The best way to picture it is to imagine a piano keyboard that's been unrolled from high notes to low notes.
So how does this map work? Well, it's all about physics. At the base of the cochlea, the part near the entrance, the basilar membrane is narrow and stiff, kind of like a high-pitched guitar string. So naturally, it vibrates best in response to high frequencies. But as you travel down towards the other end, the apex, the membrane gets wider and much more flexible, more like a loose, wide ribbon. And that part responds best to those deep, low-frequency vibrations.
And here's what that looks like in action. A high-frequency sound, like a whistling kettle, creates a peak vibration right at the base. Mid-range frequencies, which are where most of human speech happens, peak right in the middle. And those deep, rumbling bass notes have to travel all the way to the very end, to the apex, to create their peak vibration. Every single sound you hear is physically sorted and mapped to a specific place along your cochlea. It's incredible. Please like and subscribe for more custom medical videos made just for you. Now, moving on to the next segment.
So when you step back and look at this whole thing, this tiny, intricate, active engine, it really is a masterpiece of biology. It's what gives us our rich, detailed sense of hearing. But, and this is a big but, it's also incredibly delicate. It's vulnerable to damage from loud noise, from certain drugs, and just from aging. And this leaves us with a critical question for the future. Now that we know how it works, how can we do a better job of protecting this fragile mechanism? And maybe, just maybe, could we one day harness this understanding to actually repair it when it breaks?